Most of the blood tests need several hours to give out the result, and the common techniques are ELISA and Western blot. In our project, we apply gold substrate as the carrier, attach antibody on the gold surface, drop a sample on the surface, then heating with a certain intensity laser on the bottom as a stimulation. The heat fluxes from laser create an eddy current in the test sample and accelerate the binding rate between antigen and antibody. To start our experiment, the first step is to clean the gold substrate with organic solvent and distilled water. The second step is applying DSP process to attach antibody on the gold surface, which takes more than 10 hours soaking. In order to attach PDMS matrix on the gold surface firmly, we use UV ozone plasma sterilizer to clean PDMS matrix. Then we put the matrix on the gold substrate carefully. After that, we need to check and make sure they are fully attached. Before starting the experiment, we also need to record the actual laser intensity and locate the strongest intensity spot. The measured data was shown in this table. In this graph, it shows the actual laser intensity is about 40% of the value displayed on the device. After all of the preparation, we add sample in the well of matrix and cover with small glass chip to prevent evaporation during the heating process. We test each cell with control laser intensity and time span. Blue light was applied to trigger the sample emitting green fluorescence, and then we can capture the image. This image shows the fluorescence variation with different laser power. This image shows the fluorescence variation with different time spans. We can see the color tends to be more green with longer time span and higher laser power. From the pictures of fluorescence intensity, the laser power and time span both have positive effect to the antigen-antibody binding. At 110 mW and 30 minutes, the fluorescence intensity is almost 3 times stronger than the sample without laser stimulation.
In expectation, the fluorescence intensity should increase with the time span and the laser power. However, according to the result, it doesn't exactly follow our expectation. For example, the intensity at 0 mW 20 minutes is lower than it at 10 minutes. Furthermore, at 10 minutes and 20 minutes, the intensity in 80 mW are lower than it in 50 mW.
So far, the reason caused this phenomenon is unknown. However, right after the experiment, we have seen some liquid between the matrix and the substrate. It might be the reason that some wells were polluted by other wells. It might be the reason that the matrix does not attach on the gold substrate perfectly to isolate each well during the whole experiment. This issue is still necessary to investigate further.